Hello biology. Today we're going to talk about RNA and protein synthesis. This is chapter 13. This is the last chapter we'll go through before we actually take our final exam. We won't test over it, but we will have a short quiz that goes over the 13 material. So to start us off, we'll talk about RNA. And RNA is another nucleic acid. So if you can remember from chapter 12, DNA was also a nucleic acid that was double-stranded. Its nitrogen bases were cytosine, guanine, thymine, and adenine. And we had a sugar that was deoxyribose. Well, RNA is a little bit different. RNA is also made up of nucleotides, so it's made up of the nitrogen base, the sugar, and phosphate group. However, the sugar that makes up that RNA is going to be ribose sugar instead of deoxyribose sugar. So remember, the D in the deoxyribose stands for that DNA part. So the R in the ribose sugars is why we use the RNA. RNA is single-stranded, unlike DNA, and it contains uracil instead of thymine. So uracil will bind with adenine rather than thymine in RNA. There are three different kinds of RNA that help with the information to transfer protein synthesis, so you will be responsible for these three different types. Our first type is our rRNA, and these little letters in front of the RNA are going to help you a lot because they're going to be the start of the word that describes it. So rRNA we call also ribosomal RNA. That's going to combine with proteins to form ribosomes, so it's pretty easy. We have tRNA, and we call that transfer RNA, and that's going to match with mRNA codon to add correct amino acid sequences during protein synthesis, which we will go into much more detail in a little bit. And then we have our mRNA, so we have our messenger RNA, and that's going to carry the code from DNA to the ribosomes. So in protein synthesis, we have transcription of RNA. And if you can think of transcription, you can think of it in a way that, what does it mean in a literal sense, or in an everyday way? So to transcribe something means to rewrite something down. And that's basically what's going to happen in protein synthesis, or the first step. So in transcription, segments of DNA serve as a template to produce a complementary RNA molecule. So we're just rewriting or transcribing the message from the DNA and putting it into an RNA so it can be taken outside of the nucleus. So you can see here we have our template strand of DNA, and that's going to be shown in blue, and it just unwinds a little bit, and that's RNA polymerase that does that. But then our RNA strand is being created as a complement to that DNA strand. So we're rewriting the message in the DNA so that we can pull it outside and use it within the cytoplasm. Because remember, if we take DNA outside of the nucleus, something could damage it, and then that's the only copy that we have. Transcription. In transcription, the enzyme called RNA polymerase is what's going to separate the strands of DNA and then use one strand as a template to assemble the RNA. So similar to DNA replication, we had DNA polymerase. Well, because we're making RNA, you have RNA polymerase that separates the DNA strand, so that's seen right here, and adds the complements. So this is our RNA strand. So just remember that RNA polymerase is what makes RNA, DNA polymerase is what makes DNA. So how does RNA polymerase know where a gene starts and where it stops? So we have so much DNA, we have so many nucleotides. How do we know when to start to code for a gene and when to stop to code for a gene? Well, the enzymes, so the RNA polymerase, will bind to a place with specific DNA sequence called a promoter. So it's going to promote transcription. Promoters tell RNA polymerase where to start, and signals at the end of the gene code cause for transcription to stop. So there's promoter sequences and there's stop sequences that tell RNA polymerase to begin and stop. So RNA requires editing before use. So you can see here, this is everything that has been transcribed. So we have transcription happens. So we have, this is our DNA sequence. Then we have transcription happens. So this is going to be our mRNA sequence. And in our mRNA, we have both exons and we have introns. Well, introns are actually going to be removed, so that we only have left our exons, our expressed DNA sequence. Now, why waste it? Why spend energy making a large RNA and then throw away different parts? Well, we have some theories about this. This may allow some genes to be used in different ways in different kinds of cells. It may have a role in evolution, allows small changes in genes to have a larger effect. So there's a couple different reasons why we think this happens. Not quite sure yet. So DNA stays safe in the nucleus, like we said before. Transcription has to happen in the nucleus, so where we go from DNA to RNA. So remember, we're just rewriting that DNA sequence. And then translation, where we then speak. So when we translate that message, so we're turning it into a usable form, we're turning it into our protein, we call that translation, and that's going to take place on the ribosomes in the cytoplasm. So the blueprints of the master plan are carried to the building site. So the RNA are your blueprints, it's carried then to the contractor, the ribosome. So how can just 4 bases give directions to make 20 different amino acids? The message is actually read in a group of 3s. We call those group of 3s codons. So here's our regular DNA sequence. We would actually break this up. So we would count by 3s. 1, 2, 3, break it apart. So like what you see here. And then 1, 2, 3, break it apart, and so forth. So that first codon, these 3s, are going to be serine. The next one is histidine, and the last one glycine. And codons represent different amino acids. There's 64 different codons. Some amino acids have more than one codon, and we always start reading our RNA sequence at the codon AUG. And then there's 3 codons that will actually stop. So looking at this, it can be kind of confusing at first, but let's say we want to find the AUG and we want to define what amino acid that signifies. So you always start in the center here. Okay, so we start with our A, that's our first letter of the codon. The next one is U, so then we would go here to the U. And then the next one is G. So methionine is always going to be our start codon. Well, we can also find our stop codons by just following this around. Here's one. Here's a stop codon, and if you follow this in, it would be U, G, A would be the stop codon. Another one would be U, A, or U, A, G. So looking now at our tRNA, so if you remember from the beginning of this video, we talked about the different types. Here's our tRNA, and tRNAs have anticodons. So remember, an mRNA has a codon, so you see your A, U, and G. tRNA will have your anticodon, the U, A, C. So U will always match up with A will always match up with U, G will always match up with the C, because remember, we're dealing with RNA and not DNA. So the anticodon matches up with the codon on mRNA, and each tRNA carries only one kind of amino acid. So that's this part right up here. So when this matches up together, we know that that amino acid is going to start our protein. So in this picture, it just describes basically the translation process. So what happens is we have transcription happen in the nucleus. Our messenger RNA, so our mRNA, is carried out to the cytoplasm and is then connected to a ribosome. So that's what this is back here. So that's our ribosome. And then what happens is this kind of works as almost a little factory where tRNAs are going to come and they're going to attach. Once they find the AUG strand, the start codon, tRNAs are going to begin to attach, and so methionine will attach here, and then the next codon we find here, the tRNA will attach there, and then other ones will start to get in line. Well, once we've attached that one, what happens here is the amino acid is now gone from that tRNA, so then the amino acids actually combine, so the ribosome will combine the amino acids together as it shifts down the mRNA sequence. So the ribosome shifts down this mRNA sequence and continues to add the tRNA to the mRNA. So you can see that here. So these amino acids are added as tRNA then leaves. So you can see we have a long chain, and we call that a polypeptide chain of amino acids that will eventually form our protein. Once we hit the stop codon, the ribosome will break apart. So you can see here we have two different colors. We have a small part of the ribosome and a larger part. Those will break apart. The mRNA sequence will go away, and we have our polypeptide chain. So a chain that will eventually make up our protein. So proteins are the connection between the genetic code in the DNA and how that gene is expressed. So a gene that codes for an enzyme or a protein to make a pigment can control the color of a flower. A gene that codes for an enzyme or a protein adds carbohydrates to glycoproteins to produce our blood type. So here's our AB blood type. Enzymes catalyze and regulate chemical reactions, so proteins build and operate all cell components. So genes are responsible for making proteins. Proteins are responsible for basically all the biochemical pathways that take place within the cell. So when we are making DNA, going from DNA to DNA, we call that replication. So we're just making DNA. When we go from DNA to RNA, we're transcribing the DNA message. We're using transcription. When you go from RNA to protein, that is translation. We're translating that message into a usable form. So here's just a concept map that might help you, and it would be good to add these into your notes. We have RNA can be three different types. So if you remember, we have messenger RNA, ribosomal RNA, and transfer RNA. The messenger RNA can also be called mRNA, and it functions to carry instructions. So remember, it carries instructions from DNA to the ribosomes. Ribosomal RNA is also called rRNA, which functions to combine with proteins. So combines with proteins to make up our ribosomes. And then transfer RNA is also called tRNA, which functions to bring amino acids to the ribosome. And that's it for chapter 13.1 and 13.2.